So welcome, ladies and gentlemen. On behalf of Select Bio, we're delighted to welcome you. We're honored that the CEO of Spectradyne, Jean-Luc Fréquin, will be leading the discussion today. And Jean-Luc has a PowerPoint presentation and some excellent videos that he'll be presenting. And then we'll be engaging with a panel discussion. And then all the audience is welcome to participate as well. So we'll turn it over to Jean-Luc, and thank you so much for your participation today, ladies and gentlemen. Welcome, Jean-Luc.
Thank you, Nal, and welcome everyone to this exciting event. Thank you for joining us. Today we're going to unveil a cutting-edge new technology we've developed at Spectradyne and show you the powerful ways it will help you to do better science faster. We'll start by reviewing the context for why we're all here today, and then jump into the details of the ARC particle analyzer. We'll summarize the key areas of impact for this new technology, and then demonstrate how the ARC solves specific analytical challenges with a few powerful measurement examples. We'll follow with an in-depth demonstration of the technology before introducing our invited panel of experts from the field who will share their take on the state and future directions of biological particle analysis. Finally, I'll wrap up by detailing the enticing fall promotion Spectradyne's running in celebration of the release of the ARC, and let you know how you can get in touch with us to learn more about those. Let's get started. Ultimately, we're all here because we're doing some kind of nanoparticle-based science. And as we know, applications of nanoparticles are growing rapidly. Witness, for example, the LNP-based COVID vaccines, new gene therapies, particle-based diagnostics, and regular new discoveries of the important roles nanoparticles play in fundamental biological processes. For these nanoscale materials, particle size and concentration directly dictate their behavior and their performance. Size and concentration are critical parameters that determine particles' bioavailability and biological access, and ultimately, in the case of a therapeutic, a drug's dose, safety, and efficacy. Finally, the ability to identify specific phenotypes in the analysis is critical for ensuring accurate and relevant results, particularly since most relevant biological nanoparticles are present in complex, heterogeneous samples like cell culture media or raw biofluids. Being able to know that the particles being analyzed are the ones you're interested in is extremely important. But as you all probably recognize, most technologies currently in use fail to deliver on one or more of these fronts. Let's start with these two well-known technologies. I've included dynamic light scattering because it's still fairly common in nanomedicine applications, even though it doesn't provide any information about concentration or phenotype. Nanoparticle tracking analysis has historically been a workhorse in the EV world, but its limitations are increasingly well understood. Basically, its accuracy depends on the sample being measured in a way that makes it hard to properly analyze unknown samples. The landscape starts to get a little more interesting with the fluorescence-based particle analysis technologies. Fluorescence NTA still suffers from the same fundamental limitations as regular NTA, though, basically a variable limit of detection. Plus, practical complexities like photobleaching make quantitative single particle fluorescence difficult. SP Iris with fluorescence is a powerful technology for size and phenotype, but is not quantitative for concentration. It's just relative counts. And flow cytometry is a well-developed method for large particles in cells, but it hits the diffraction limit trying to use it to detect smaller particles. This means it's hard to detect those ones, and you have to assume optical properties of the particles to calculate their size, so it becomes a challenge to analyze heterogeneous samples. Spectradyne's core technology, Microfluidic Resistive Pulse Sensing, or MRPS, delivers accurate nanoparticle size and concentration. But the NCS1 works best in applications where the sample has been purified, when the particles of interest have a standout size distribution like a virus, because the MRPS technique alone doesn't identify what particles are being analyzed. So in this technology landscape, there still remains a critical need for technology that can accurately measure the particle size, concentration, and phenotype. And so without further ado, allow me to introduce formally the solution. All right. Spectradyne's ARC particle analyzer is a new and groundbreaking technology for the analysis of biological nanoparticles that builds on Spectradyne's proven microfluidic resistive pulse sensing technology, or MRPS, and now delivers simultaneous single particle fluorescence-based phenotyping. The ARC specializes in quantitative fluorescence measurements using native NIST traceable units. And as we'll see today, its sensitivity is state-of-the-art in up to 3 optical detection channels. Particle concentration and size are measured electrically by MRPS from 50 nanometers and up, ensuring accurate measurements and ultimately complete subpopulation analysis in even highly complex samples. As we'll see in the demo today, the instrument is much faster and easier to use than conventional technologies and requires just 3 microliters per sample. Importantly, the ARC is compatible with commonly used standard staining protocols that have been developed over many years for flow cytometry. This powerful combination of accurate particle size, concentration, and phenotype will deliver benefits in a range of interest areas. For extracellular vesicles, the ARC delivers fast and accurate phenotyping to speed your research. If you're working in a nanomedicine application, the ARC will allow you to accurately measure the dose of your therapeutic and characterize surface and cargo markers even in complex formulations. In virus and gene therapy applications, the ARC delivers specific viral titer in minutes without requiring tissue culture. And finally, in diagnostic applications, Spectradyne's ARC enables the identification and quantitation of new biomarkers at the nanoscale where other technologies have not been able to perform. Let me illustrate these deliverables with some measurement examples now. First example I'll show you is an extracellular vesicle analysis in which we quantify a subpopulation of fluorescent EVs. This plot is showing us the measurement of this sample, and on the y-axis we're showing particle concentration, and on the x-axis is particle diameter. You can see there's a log scale in the concentration in the y-axis, and this particle size distribution shows all the particles in the sample as measured by MRPS. So this would be the same as if you measured it with one of our NCS1 instruments. Typically, it shows a broad size distribution that's very common for this kind of sample, with an approximate power law dependence of concentration on particle diameter. But now we're measuring it on the ARC. And so this is an example of the raw data that we get from it. And you can see 3 measurement traces here in versus time now. So this is the raw signal versus time. In blue is showing the MRPS signal, where we're detecting a particle. This downward spike is a particle detection by MRPS. And simultaneously with that now, we're also measuring quantitative fluorescent signal from each particle in 2 channels. So now when we come back to this distribution, we've measured all the particles, and we can also call out specific subpopulations that are FITC positive or PE positive. In this particular sample, there's a clear fluorescence population shown in green that are FITC positive fluorescent EVs as a subpopulation of this much higher background of non-fluorescent particles, and almost no PE positive particles in this sample. As always with Spectradyne's technology, the data is quantitative, and so we can measure the concentration over any size range here, and we immediately see that the FITC population is about 8% of the overall particle concentration in this selected size range that we've shown here. We'll dive into an example like this in a little more detail in the demo coming up shortly. The second example I'd like to show you is from a virus application or a gene therapy application. And what we're measuring here are GFP expressing murine leukemia virus, or MLV. When we measure it, this is the measurement result we see. So again, we've got concentration on the y-axis and particle diameter on the x-axis. And in blue, we're showing all the particles as detected by MRPS. You can see there's a well-defined peak from the virus particles in the sample, and it's sitting in a background of other non-fluorescent particles here. And we now know that for sure because on the ARC, we're measuring, we can call out just the FITC positive particles, the particles that are fluorescent in the FITC channel. These are GFP labeled. And you can see that this background, now we know, is non-fluorescent particles in the buffer or other parts of the sample preparation. Maybe they're leftover bits from the cell culture media, for example. Again, we can be quantitative over any size range here. And in this case, what I'll do is we'll ask the question, how bright are these particles? So we can put a gate around the fluorescent virus particles and select only those particles. That's what's shown here now. And now this plot here is showing the concentration versus on the y-axis versus brightness in equivalent number of reference fluorophore units in the FITC channel. So you can very clearly see that the mean brightness of these virus particles are about 760 equivalent number of FITC fluorophores. We can measure the concentration directly, and you can see we've measured over 500 particles in this example to get the data that we need. This measurement took only 3 minutes, and delivered the particle size, which is about 150 nanometers in diameter, the virus concentration, about 2e⁸ particles per mil. We now know that about 71% of the particles on this size range are FITC positive, fluorescent, and we've been able to quantify the brightness of these virus in translatable NIST traceable units. Finally, I'd like to touch on the question of how sensitive is this technology. So let's run a set of reference beads so that we can assess that directly. What I'm showing you here now is a measurement of a mixture of 2 polystyrene beads. They're 180 and 200 nanometers in diameter. And we have had these particles cross-calibrated on a flow cytometer, because they're big enough for that, to become our spike-in standards at Spectradyne. So we know that the 180 YGs beads are as bright as 964 FITC molecules, and the 290s have appropriate values also. This measurement of the 180 YGs for the PE channel, they were too dim for the flow cytometer to accurately obtain an assignment for this sample. So let's look at these in terms of brightness now. Starting with the FITC channel on the left here, now we're showing the concentration versus the brightness in the FITC channel in terms of translatable units. And you can see clear populations for the 180s and the 290s at their expected brightnesses and we can look at where the noise is in the system. Based on this measurement, we can see that the detection limit in the FITC channel is somewhere around 20 ERF, right? Approximately 20 ERF. We can do the same thing for the PE channel, and when we do that, we attain a detection limit of about 1 ERF unit. So these sensitivities are very high and compete very strongly against conventional flow cytometers that are on the market right now. So let's see it in action, see how easy it is to use.
Introducing the Arc Particle Analyzer by Spectradyne. Meet Nak Do, our senior application scientist here at Spectradyne. She will be using our Arc Particle Analyzer to quantify a subpopulation of fluorescent extracellular vesicles in a complex mixture. Specifically, we'll measure the concentration and size distribution of GFP-expressing vesicles in a much higher background of non-fluorescent vesicles. Why did we pick this measurement as an example? Because no other technology can successfully execute it. These particles are sub-100 nanometers diameter, weakly fluorescent, and don't scatter light strongly, so accurate concentration measurements by flow cytometry or fluorescence NTA are not possible. These particles also have heterogeneous optical properties, so a single refractive index for all particles cannot be assumed to determine their size by flow cytometry. And techniques that require immobilization of the particles on a surface take a long time and aren't quantitative for absolute concentration. Only the ARC is capable of accurately performing this measurement because it uses MRPS to directly measure the size and concentration down to 50 nanometers, while simultaneously measuring single particle fluorescence to identify the subpopulation of interest. Let's get started. First, load just 3 microliters of the sample to be analyzed into our microfluidic cartridge. Then simply open the cartridge interface on the instrument, put the cartridge in, and close the lid. Then we can start getting the instrument and cartridge ready for measurement. A key feature of the ARC is that the focus and alignment of the excitation beam and collection optics is completely automated, without separately having to run a dedicated sample. Simply press autofocus and align, and wait for the process to complete. This feature means a much simpler workflow than for flow cytometry and ensures repeatable results from run to run. This image is showing us that the excitation beam is perfectly aligned directly over the MRPS sensing constriction in the microfluidic cartridge. Once the optics have been aligned, it's a single click to begin acquiring data. Particles are analyzed in 10-second acquisitions, and the raw data is displayed after each is complete. Let's zoom in for a closer look. Particles are detected electrically using Microfluidic Resistive Pulse Sensing, or MRPS. The MRPS measurement is shown in blue, and this downward spike is an example of a particle detection event. You can learn more about how MRPS works on our website. Now with the ARC, we're not only counting and sizing particles, we're also simultaneously measuring each individual particle's fluorescence. This particle in particular generates a large amplitude signal in the FITSI detection channel and a smaller amplitude signal in the PE channel. Each acquisition is processed in real time by our auto analysis engine. The auto analysis engine also offers a variety of ways to view your data in real time. The live CSD shows the particle size distribution as measured by MRPS. So this is showing us the distribution of concentration versus size for all the particles detected so far. The live CSD also shows us the particle size distribution for the subset of particles that exceed a brightness threshold in each fluorescent channel. We can immediately see from this that there is a well-defined subpopulation of particles in the sample that are fluorescent in the FITSI channel. These are the particles we're looking for. How can we be sure these are real particle events? The ARC uses MRPS, a non-optical technique, to count and size particles one by one without making any assumptions about particles' optical properties. This means we know for sure that every fluorescent particle is a real particle, and not an artifact of using an optical technique to detect particles smaller than the wavelength of light. The auto-analysis engine permits a variety of parametric scatter plots to be viewed in real time, or as a function of time. This particular view shows fluorescence amplitude in the FITSI channel versus true particle diameter. Remember, no assumptions have been made about the particle's physical properties in order to calculate their diameters. And this is what we're after. The particles of this population are clearly fluorescent. These are precisely the GFP-labeled EVs we're looking for. You can see in this plot that the majority of particles in the sample are non-fluorescent. The fluorescence amplitude of these events are below the limit of detection, and we can see them being detected in real time. After counting sufficient particles to ensure statistical confidence in the concentration measurements, in this case, a few thousand particles, we can end our run. Today, we've shown you the simplicity and power of Spectradyne's ARC particle analyzer. The ARC delivers accurate measurements of particle size, concentration, and simultaneous single particle fluorescence at the nanoscale, all in an easy-to-use desktop system. Reach out to our sales team to learn more.
All right, the ARC is fast, easy to use, and powerful technology. Let's shift gears now for another exciting portion of the event, a live discussion with our panel of scientific experts from the field. Let me introduce them to you now. Dr. Jennifer Jones is a Stadman investigator in the Laboratory of Pathology at the NIH National Cancer Institute. Dr. Ken Whitwer was supposed to join us, but apparently is having technical issues. Hopefully he can jump in a little bit later. Associate Professor of Molecular and Comparative Pathobiology at Johns Hopkins School of Medicine, and Dr. Edwin van der Poel, scientist at the Department of Biological Engineering and Physics at Amsterdam University Medical Center. It's important for me to say that at Spectradyne we put scientific rigor at the top of our priority list, and so we've invited these panelists for their expert opinion about the scientific challenges in the field of biological particle analysis, but their participation does not represent an endorsement of any of Spectradyne's products. We just hope to have a healthy discussion of the science involved, right? So welcome panelists, and thank you for joining us today. Maybe we can, do we have communication? Dr. Jones, can you hear me?
I can. Can you guys hear me?
Yes, great. Okay, and Edwin, you're on too?
I'm here.
Great, excellent. You're a little bit quiet, so speak loudly. Great, so maybe we can start with you, Dr. Jones, and you can give us your thoughts on this first question. So why would you say that accurate concentration of biological particles is important?
That's a key question for us in the translational sciences, as well as those who are studying these things in the physical sciences. You mentioned that size and concentration are things that we need to know in terms of dosing of nanotherapeutics for their safety and their efficacy. But a lot of people are also interested in these biological nanoparticles in biofluids for their information that they can convey to us relating to health or disease. And since these are diagnostics, it's really important that when we have a number, that there is accuracy to that number, meaning that it reflects the truth of what's really there. And with these nanoparticles, the challenge to seeing the truth of what's there is the detection limits of the instrument. So for example, if I had a patient and I needed to know how many tumors does this patient have, if I did a CT scan of just the thorax and I found 2 tumors, it might actually be very inaccurate for me to say there are 2 tumors there. But if we did a whole body scan, we might see that there are actually 6 or 8 tumors. So, you know, when we're actually doing diagnostics, it's really important that the accuracy reflect, that the measurements reflect the truth of the situation. And you've helpfully emphasized in this how you're able to establish the limit of detection. So when we report with measurements like this, where you can assess a limit of detection on a calibrated scale, then you can report truly accurately in this measurement, in this range. This is what we see. So that's really helpful to add that accuracy.
Cool. That's a helpful perspective, especially given that the importance of these measurements. I mean, people making measurements for diagnostic decisions, you know, have real impacts in the real world. Edwin, do you have anything you'd like to add to that?
Yeah, I think it was a quite complete story. I mean, it needs to be accurate, I think, to improve the general reproducibility crisis in life sciences. And yeah, I happen to work with metrologists, and they actually shiver a bit from the question, could you please determine the concentration of these nanoparticles? So that I think shows that determining the concentration in an accurate way, a way where you roughly know preferably just know the uncertainty of the measurement, is very difficult. No.
Right. If I could translate what you were saying, Dr. Jones, the tumor example, you're essentially saying that the thorax measurement, is defined by the limit of the area where it was the limit of detection is the thorax, right? And we could also say, so in our kind of measurement, the analogy would be that within this size range, these are the number of particles that were counted, right? And if we're talking about counting just the fluorescent particles, then it's also important to state how bright the brightness limit of detection also, right? Mm-hmm. Because that's another bound on the measurement, right?
Mm-hmm. Yeah. With this measurement, with this assay type, with these limits of detection, this is what we see.
Right. Cool. That's very helpful. Edwin, I know you're very big on the quantitative fluorescence question. So can you take this one? In your opinion, why would you say it's important to be quantitative with the fluorescence measurements associated with flow cytometry or this technology?
Yeah, all right. Now let me start with why fluorescence is important. I mean, body fluids or other fluids containing cells often just have a very complex mixture of particles, right? So for most applications, only the concentration of a specific subset of these particles is relevant. So the detection method should be able to identify these particles of interest, and immunofluorescence labeling or other ways of fluorescence staining are an established way to identify biological particles. So while immunofluorescence labeling or staining procedures are broadly applied to identify cells, it can also be used to study submicrometer particles. Now, why quantitative? And I think, yeah, the answer already popped up in a little bit in the previous question. Submicrometer particles in biofluids are heterogeneous, and therefore they differ in the available binding sites. So if you would make a distribution of the fluorescence intensities, you get a very broad distribution. So if you want to start comparing this information to other labs or aim for a reproducible assay, it's important that you quantitatively know the intensity range wherein you are measuring. So that's the first. And the second one is rather speculative, but yeah, of course, the quantitative information on the fluorescence distribution may also have a yet unknown clinical potential, but of course that remains to be investigated.
Sorry, I didn't understand your last point. With the speculative question, say it again.
Now, the shape of the fluorescence distribution of particles in biofluid could be interesting as well. So if you could quantify this shape, and again, due to the quantitative manner, you can start comparing it and reproduce its data. I think that's very interesting to investigate.
Oh, cool. One thing we haven't talked about in terms of quantitative fluorescence yet is trying to be quantitative about the number of like surface markers, for example, and trying to get at the biological implications of that. Care to comment on that idea?
Yeah, I think preferably, actually, I think most people in the field are not interested in how quantitative you can determine the fluorescence itself, but they are interested in the end in how many receptors did I bind with my label, right? Or even better, how many receptors does my or particular proteins does my particle have? But the step to that answer is, of course, a quantitative fluorescence signal. Because if you know how efficient your label binds, etc., you know this biochemistry and biophysics thereof, you could make the step from quantitative fluorescence to an estimate of the number of proteins present on your particle.
Is that something that's done with conventional flow cytometry for larger particles? Or the numbers too big for that?
It's done, but I mean, this is very, it's very specific. So what I rather see in the literature is that people stay with units like ERF and MESF just to have their assay reproducible. And to go to the number of receptors, you really have to go into details of your own assay and reagents that you are using.
Right. Any thoughts to add, Dr. Jones? Yeah.
Yeah, I'd love to ask, and I think I know the answer to it, but given what Edwin was saying about the heterogeneity of the material that we're looking at from biofluids, presumably because you're analyzing these at a single particle level, there will be an opportunity or an ability to gate populations and interrogate the heterogeneity in that way, in a similar way that we do with flow cytometry, to say, take the largest 10% of the particles detected and see if the relative intensities of, say, FITC or GF or PE are equivalent to, say, the distributions of those fluorophores on a mid-range size detected by MRPS. So I think the opportunities for doing gated analyses within this is going to be exciting as a first step in exploring the heterogeneity.
Yeah, cool. I mean, we didn't explicitly show gating in the data that I've showed today, but it's implicit in the analysis because, for example, that subset of fluorescent GFP EVs amid a sea of background non-fluorescent ones, we're essentially isolating those to measure, based on their fluorescence, to measure their concentration and size distribution. So it's definitely possible, and that's an exciting idea for what could be done with this. That's great. I was going to pose the next question to Dr. Whitwer, but since he's not here, we can ask you guys. Obviously, it's great. So, what would you say? You're both very familiar with the technology landscape for nanoparticle analysis. In your mind, what are the current roadblocks or needs for characterizing biological nanoparticles in your mind? What would you like to see come in the future?
That was to me or to Jennifer? Sorry, I missed it.
Sure. Well, you unmuted first, so go ahead.
All right. This is a very broad question, and of course very experiment specific. What I think are the current roadblocks is it's perhaps a mixture between detection and isolation, because if you look at the field, there are two major streams, at least the field of EVs, but I think that accounts for many fields. So either people isolate particles and they apply omics kind of methods or other ensemble methods, or they do single particle detection. Now, in the single particle detection, you have now several options since about a decade. Like flow cytometry, like MRPS, like NTA, and a few of these technologies become more and more sensitive, and they are now below 100 nanometers, right, with their sensitivity. And I think one major hurdle for the future there is to gain sufficient statistical information, because in some body fluids, the number of particles is rapidly increasing with decreasing diameter below 100 nanometers. So such single particle detection methods need to be very fast in the future, or be combined with an isolation method. And then the question is, what is the best interplay between that isolation method, which still might have to be developed, and this technology?
Right.
Yeah,
So,
I would echo
ahead.
exactly what Edwin was saying, and say that the roadblocks that we face arise from the smallness of the size of these particles and their heterogeneity, and the fact that all of the instruments that we're using began at first as instruments adapted from the larger cellular space. And it's only now with dedicated instruments that we're starting to have applied capabilities for these small particles. So I think, as the field continues to grow, and we identify that in these biofluids, it's not just extracellular vesicles, but there may be other extracellular particles that have been classified as exomeres or supermeres. And in this mix of this wash of other proteins and macromolecular complexes, how do we characterize those even smaller than 50 nanometers? So I think it's really encouraging to see the field continuing to step forward in this direction, and to be able to have an instrument that has orthogonal assessments with the same sample. Orthogonal measurements help us quite a bit in assessing the reality of what we're seeing and the attributes of the particles we're studying.
That's great, and I'm glad you mentioned the smaller particle stuff, because that's Spectradyne's next focus area, is to scale our technology to even smaller particles. So now that the ARC is available and on the market, we'll direct our efforts in that direction. Well, thank you both for your time and participation in this. Any closing remarks from either of you?
I didn't have anything particular in mind, but I would like to thank you for inviting us. Pleased to meet.
Thank you. I agree. I think there's some great questions in the discussion in the chat as well.
Okay, great. Yeah, we should have time to get to some of those at the end. That's great. All right, thank you very much, both of you. All right, so today we've introduced you to Spectradyne's new product, the ARC Particle Analyzer, which delivers accurate particle size, concentration, and phenotyping. I hope you're as excited as we are about it now at Spectradyne, because we're really pumped over here. We've been working on this for a long time, and it's exciting to launch it for real into the world. I'd like to acknowledge also that the development of this technology was supported in part by an SBIR grant from the NIH, the National Center for Advancing Translational Sciences. Now, let me wrap up by sharing a couple of special promotions we have running in celebration of the launch of this exciting technology, and then we can get to a couple of the questions in the chat after. So we have instrument promotions running through the fall. ARC early adopter deals, especially in September, 25% off the list price. And we are offering special trade-in deals if you already have an NCS1 through the end of November. At the same time, we're also discounting our NCS1 instruments, so you can save on those as well. Or if you know somebody that needs just the MRPS portion of it, still a lot of utility for that technology, as we talked about before. Through November 30th, we'll be offering 20% off the list price of NCS1. To learn more about that, please contact our sales team or visit our website to learn more. We have a lot of more information coming on the website today at the same time as this launch event. Thank you very much for joining us today, and please stick around for the Q&A. Let me see if I can get at some of these questions in the chat.
Also, ladies and gentlemen, and Jean-Luc, if people would like to ask questions in real time, you're welcome to raise your hand and we'll unmute you so Jean-Luc can address them right now?
Yep, sure. So I see one in the chat. When we're looking at the axis of fluorescence signal against the size axis, can you translate the signal directly into ERF? So can you directly plot the signal on an ERF calibrated scale so that we know how to close the limit of, how close to the limit of detection the EVs are? The answer is yes. All of our data is by default actually plotted against an internally calibrated ERF scale. You can validate that, for example, with a spike in of a known brightness in your sample, just the way we do for concentration if you want to. And that's very easy to do in the software, definitely. There's a question from Jeffrey Clogston at the NCL, the National Nano Characterization Lab. Hi, Jeff. Jeff asked, since fluorescence measurements per single particle is possible, can you look at drug loading and extent in nanoparticles containing a fluorescent drug? For example, say which nanoparticles have no drug at all versus yes, there is drug present in it, and then how much drug is contained. That is an excellent question, and the answer is yes. And we would love to demonstrate that with samples, hopefully, well, potentially from you or with anyone. So let me make a general solicitation for samples. We would love to demonstrate our technology of the ARC on your sample specifically. So contact our sales team to figure out how to send those samples to us. But Jeff, yes, that's a super exciting application of this technology that we'd like to explore more and demonstrate to you personally. So please reach out. That's great. Let me see. There's a question, does the ARC use the same cartridges as the NCS1? And the answer is basically yes. So they're interchangeable. So if you trade in your ARC or trade in your NCS1 for an ARC, you'll be able to keep your cartridges and use them with the next gen technology. There's another question in here. Can we get a distribution of particles that are double or triple positive? The answer is yes, because we can do fluorescence detection in up to 3 channels at once. So we could characterize positivity in, say, a FITC configured channel, a PE channel, or an APC configured red, deeper red channel also. Right, building off the drug loading question, this is from Chris Haycock. Thanks for joining, Chris. Are fluorescence measures limited to external loading, or can internal labels be measured as easily as well? We have not, actually the reference particles, the polystyrene particles that we use are internally labeled. So certainly there's some contribution from the internal fluorophores in the sample. My expectation would be, because the particles are small and for sure the excitation light will penetrate into the particle, that the answer is yes. So, but we'd love to check that. There's a question, is the ARC able to measure a single fluorophore molecule, or is that below the detection limit? Well, so the data we showed for the PE sensitivity, because PE is quite a bright fluorophore, right? Show that in principle, yes, we should be able to detect the fluorescence of a single particle or a single fluorophore. We're right at that capability now. So send us a sample that we can validate it on particles. That would be great. There are lots of questions about GMP compliance, and so let me just address those generally. So GMP compliance, as I understand it, is a validation of a technology within the context of a particular workflow. And so no technology itself is GMP, validated, compliant, but given a protocol, then it can be validated as part of an analysis process. And so we would be happy to, that's, by nature, that is application specific, but we're eager to work with you to develop that protocol also. So please reach out, that would be great. Lou, do you see any other questions in here that we should get at?
Yeah, there's one here from Michael Evender. It says, I guess, let's see, I guess you are using traditional cytometry fluorophores. Have you looked into anything into how to avoid counting fluorophore/antibody aggregates as fluorescent positive vesicles. This has been a big issue for us with fluorescence techniques for vesicles before and after filtration.
Thank you, Michael. That's an excellent question, and I'd love to answer it because one thing that's very special about this technology is that we are gating on only real particles that are detectable by MRPS. And so many fluorochromes may aggregate into very bright particles that would show up as bright fluorescence events on a flow cytometer, for example. But because they're not actual physical real particles to be detected by MRPS, above 50 nanometers volume, then we will not be seeing those and they won't contaminate the data. So that's a very powerful difference between, that's a powerful deliverable that the MRPS technology brings for the particle detection. I hope that answers your question.
Got another one here from Shiva. Hi, Shiva. Apart from getting relative percent of fluorescent EVs, would it be possible to collect the different populations of light flow sorting? And the answer is not currently. So,
right. Yeah. Future technology flow sorting at the end of this one.
Oh,
yep, I think.
Sorry, I have another question, Jean.
Yeah, sure.
So if they ask me to unmute, can you export data to FCS files, which is the flow cytometry standard?
We have a export that is not quite FCS format. That's in development, but we do have a CSV format that lists the particle events with their parameters. So that is coming for sure, and I recognize that's an important capability because a lot of our users will be flow cytometry users and used to using software like FlowJo and other flow cytometry analysis tools. Yeah.
One other question from Michael, again, it's a Mikhail or Michael, I'm not sure, but what limitations of the sample is there? Can I inject cell-free plasma directly? Does it need dilution? If I have a particle in a buffer, would it need a surfactant added? That's several questions at one, but.
Yep. Good question. So on the NCS1, which is fundamentally the same, microfluidic cartridges and getting the sample to flow through there, we know that we can run cell-free plasma pretty much neat. It contains enough proteins and other stuff in there to wet the cartridge without the addition of a surfactant. You can't run things like pure water because, A, it's not conductive. So the sample requirements are that it be conductive, like plasma, PBS equivalent or so, plus or minus, and then that there be enough of a wetting agent to allow the sample to come into the cartridge properly. And we have a variety of options available for helping develop samples that will run smoothly on the instrument.
Another question from Shiva. Have you done staining of EVs using antibodies or antibody fragments and done the analysis on the ARC?
Oh, yes. And we'd be happy to share that data with you separately. There's a limit to how much we could squeeze in the presentation today, but yes.
And
should be compatible with, all the flow cytometry standard staining protocols and things.
One last question here I've got from Peter Hauer. Is the detection confocal? And I'm not sure quite what he means with that, but I think I know what he's getting to, because then he proceeds to say, what are the chances of double events in the optical channel? Do the time resolution of MRPS and optical detection match?
Yeah, very good question. So this becomes an issue with flow cytometry where it's called swarming, right? And we can ask Edwin all about this, where a single fluorescence event in a flow cytometer may be comprised of more than one fluorescent particle going through at once, and there's really not a good way to distinguish that from each other. And so you have to do a very long dilution series to make sure that you're in the correct linear range for concentration, to make sure that you're not seeing multiple events at once. With the ARC, however, remember we're detecting particles with MRPS, and so we have a very physical measure of the particle characteristics as they go through the constriction, the size and concentration. So just like if you're running on the NCS1 and the concentration is too high, you can see that very directly in the raw data because particle events are very close to each other and in time, and they start overlapping with a reasonable frequency. So we should be able to avoid that situation pretty well on the ARC. I see Jennifer has, Dr. Jones has her hand raised, so let's.
Yeah, Edwin and I are familiar with this swarm problem.
Right.
Before Edwin popularized the term swarm, flow cytometry referred to part of that problem as coincidence. And it may be that there may be still coincidence challenges where a particle could run through the resistive pulse sensor and a fluorophore could coincidentally be detected by the detector. So the same sorts of efforts that we have to think about to reduce the amount of background fluorescence may be something that might have to be considered, will probably have to be considered with this as well.
I see. Yep, that's a good point.
I have one other question here that just came in, and it actually relates to an earlier question, so I figured we should cover it. Do you have a way to adjust for the added size of the antibodies when staining EVs? Is it noticeable? And I'll go ahead and answer that because the earlier question was related to do you have to assume that the particles are spherical? And this is part of the beauty of this system, is that we're making the measurements of the size and concentration from MRPS, which is an electrical detection, and it actually directly measures the volume of the particle, and that makes no assumption as to the shape of the particle or the material it's made out of. And we've actually shown incidences, and actually there are published papers, for instance, that show that MRPS can shows a change in size, for instance, on COVID from the spike protein. So the answer to that is yes, we would, if it's detectable, we would notice the difference in size from the added size of the antibody. But it's got to be detectable. So if it's and it's all going to be relative. So if it's a 0.1% difference, you're probably not going to see it. If it's a 5% difference, you're definitely going to see it. And
right.
Jean-Luc, you can add to that if you want.
No, that's great. I'll just point out, so if you're worried about the EVs changing size from a specific marker that sticks to them, you could spike in something of a constant size into the sample measurement as a reference. And then when you do that, then you become very sensitive to changes, but you can account for those also, right? So we can, happy to talk about that in more detail also. All right, I think we are about out of time. So let me thank everybody again, including our panelists. Thank you very much for joining us. And please reach out to us to learn more about the ARC particle analyzer. We're looking forward to hearing from you.
Thank you very much, Jean-Luc. Ladies and gentlemen, we're delighted that you participated. If you'd like to reach out, you're welcome to email us or directly to Jean-Luc and his team, and they can respond to you straight away. Thank you, and hope everybody has a great day. Goodbye.